It's my pleasure to introduce our next guest from the Center for Breakthrough Medicine's Chief Technology Officer, Avi Nandi. As a reminder to attendees, if you'd like to ask a question of our speaker, please type it into the question box under the video player, and we will try to ask it on your behalf. The format for this discussion is a fireside chat. We'll go about 25 minutes and then ask any audience questions at the end. So Avi, welcome. Thank you so much for joining us today. And maybe to start, could you provide a couple minutes of introductory comments about the Center for Breakthrough Medicines for audience attendees new to the company?
Sure, and thank you for having me. So the Center for Breakthrough Medicines is a contract development, manufacturing, and testing organization with the mission to advance the development and manufacture of advanced therapies, because we understand the importance of these therapies to the patients and the families, in which in many respects, this may be their only line of treatment. And so we are located in the King of Prussia, and we offer an end-to-end solution to advanced therapy companies that's really broken down into 4 key business units. The first is our nucleic acids business unit that offers both plasmid and mRNA capabilities. For the plasmid, we offer research grade, what we call GMP-like or Precision Pro in our shop, and GMP plasmid. And then we also have a vector business unit, a cell therapy business unit, and a testing business unit to be able to offer all process development, analytical development, manufacture, fill finish, and release testing for your advanced therapies that you need to deliver in an effective and an efficient manner. We have built out roughly around 350,000 square feet of 700,000 square foot total build out. And this does include all supportive services such as R&D services, where we have invested internally to develop technologies that we believe address pain points that exist currently in the market, but also those in anticipation of future needs, and all quality and regulatory services.
So I guess in terms of the status, can you be more specific about what types of suites are up and running and what types of suites you anticipate bringing online by the end of this year?
Sure. So we have a pretty significant build out with capabilities that are online right now, and the new capabilities that come online over the next couple of years. So from a process development, so if I start with development and then move over to GMP manufacture, from a process development perspective, we have established roughly around 10,000 square feet of laboratory space that spans between nucleic acid, viral vector, and cell therapy capabilities. And in our process development space, we perform activities from the bench scale all the way through pilot scale, which would be at scale GMP manufacturing, but in a non-GMP environment. From the testing and analytics perspective, we have roughly 20,000 square feet of analytical development and GMP laboratory spaces currently built out, and that's increasing another 18,000 square feet through the remainder of 2023. So we expect that space to come online in roughly the July timeframe. From a GMP perspective, we have roughly 8 viral vector GMP suites available that supports the manufacture of 3 high throughput lines. So this would be roughly a cell expansion upstream and downstream suite to manufacture your drug substance. And that's roughly around 27,000 square feet of space, which can produce roughly around 15 to 30 batches per year, depending on what your process looks like. And then we have the supportive fill and finish services. So we have 2 filling options in a separate dedicated suite. And there we'd be able to support formulation, fill, inspection, and packaging services. From a cell therapies perspective, we have 10 suites. This is, sorry, for GMP. We have 10 suites that we anticipate to come online in late Q3, early Q4 of this year. And then roughly once we have a capability online, we have another floor coming online roughly per quarter after that, which comprises roughly between 75,000 to 100 square feet of additional GMP capacity. And so, as I mentioned before, we roughly have around 350,000 square feet built and moving upwards of 700,000 over the next year or so.
Well, great. What you're doing out there is certainly impressive. We did have the opportunity to visit last summer. And it's clear that a key objective is to build your operation intelligently, and we continue to see you add pieces that support that. So could you discuss some of your recently announced collaborations, maybe starting on the viral vector side, your agreement with Asimov for clonal HEC-293 suspension cell lines, and also the tech transfer of UPenn's GTP adherent cell manufacturing platform?
Sure. So one of the critical raw materials in the viral vector space is your cell line. So, typically for lentivirus, for AAV, for adeno, you're using an HEK-293 cell line, and the technology is really moving from adherent platforms to suspension platforms. And honestly, there's limited options available to innovator companies for what cell lines to use. And the ones that do exist can be fairly pricey, depending on at which stage you're implementing that cell line. Typically, once you're entering the GMP space, you're entering a phase 1 trial, there can be some, fairly high cost associated with the use of the line. And then for those fortunate enough to enter the commercial space, those then continue. So we believe it's important to really enter a strategic relationship with Asimov, who then is providing us their suspension HEK-293 line to be able to offer another solution or a different solution to innovator companies that is more affordable, but without compromising on the performance of the cell line. And so we entered into a strategic agreement with Asimov to have access to their cell line, and then we offer that both internally to our clients at little to no cost, depending on the phase at which their work is at. And then also we offer it externally to clients as well. In addition, in the viral vector space, just to kind of continue on that theme, we also entered into a strategic relationship with the University of Pennsylvania Gene Therapy Program, where we have access to their AAV platform, which they have demonstrated over numerous different products that I think that has supported 4 clinical trials, which is an adherent-based platform, which really expedites the development and tech transfer of new programs that are AAV-based, where we can experience tech transfers in as short as 4 months from clinical candidate selection into GMP manufacture with a demonstrated track record for success across multiple serotypes.
Okay, and then on the raw material side, you announced agreements with Nucleus Biologics for their custom cell culture media, and with Stoic for their cell media manufacturing. So tell us how these help to add to your capability.
Sure. So you can imagine, as a CDMO, we are going to partner with a diverse set of clients. And in particular, you can imagine that the buffer and material needs are going to be critical for us to reliably source. And so it's important for us to enter a strategic relationship with both Nucleus Biologics and Stoic Bio. So in the strategic relationship that we have with Nucleus Biologics, we're in essence de-risking our supply chain for sourcing the media and the buffer that are used for our different clients, while being able to maintain a consistency of manufacture and delivery, but also the quality control of those buffers. And so oftentimes, you can experience challenges in consistency, you can experience complexity in the processes used to formulate these medias. And so partnering with Nucleus Biologics, we feel like we have really mitigated one of the key risks in our supply chain that is important to our clients, as in many cases, the logistics and the manufacture of these can be complex. Now, when speaking about Stoic Bio, they offer a unique technology called the Krakatoa. In essence, it's an on-site machine or piece of equipment that formulates your media when you provide a certain recipe to Stoic Bio. In essence, it's like a Keurig machine. We provide a formulation, they provide us, a pod, we enter it into the machine, and then it does all the formulation and hydration within that machine, and then out comes your sterile, media or buffer. And so we see a significant advantage in this because it, one, it reduces the complexity of the formulation process, right? So, for some of these medias, they have a specific order of operations that you have to go through to ensure that you meet that, that final specification. And this really removes the complexity of it, but it also simplifies the supply chain. It allows us to be able to order it, then be able to formulate on site, really in a more on-demand fashion, and simplify the storage of a lot of these buffers, which when you talk about hundreds of liters of buffer can be fairly complex. And so we're evaluating their Krakatoa technology at bench scale right now, with the future hopes to be able to scale up the technology and use it in our GMP space to then really simplify our access and formulation of media and buffers.
Well, great. So CBM is going to be able to offer services to support both cell and gene therapy development. And maybe on the cell therapy side, and specifically for autologous cell therapies, what are you doing? What technology advancements are you incorporating that you think will have a significant impact on reducing manufacturing cost of autologous cell therapies?
Sure. So we're really approaching this in a few different ways. The first one is, when a client process comes in, oftentimes there are numerous open manipulation steps. And open manipulations in the cell therapy space introduce a human element to it, a degree of variability that we see can be problematic to client processes. And so, one of the first things that we do is an evaluation of their process and look for opportunities to close it, to then really streamline the operation and have more consistent outcomes. In addition to that, we also look at reduced harvest processes. So you can see, some autologous processes span, between, let's say, 10 and 20 days. And that creates many different challenges, but mostly to the patient, it creates a risk in the time it takes to be able to receive the ship the sample, receive it, process, and then send back. And in some cases, the patient may not have the time to afford. So we believe a reduced harvest process, where you can get the process down to 1 to 2 to 3 days, affords an enormous opportunity to the patient, and is an area that we are investing in. In addition to that, we are looking at optimizing manufacturing processes and looking at how we can sort of redesign facilities to create a more high throughput manufacturing. So, for many autologous processes, there's sort of a limited set of equipment that is used over an extended period of time. And we're really trying to rethink how equipment, how rooms, how operators are utilized to be able to optimize the throughput of a manufacturing facility by breaking up a process into individual components, where the operation on that can be higher throughput in a manner, then affording overall a higher throughput within any given space, or higher throughput on a per square footage basis. The next thing that we're doing is we're implementing a fully digitized solution to manufacturing. We think this is one of really our key components in our partnership with Autonomous and an EBR. And we believe that there are numerous, let's just say, efficiencies or opportunities to improve both the manufacturer and reduce errors in the process by implementing a system like that. And on the similar respect, we're looking into automation as well. So how could you even further remove the human element by automating the process? And the final element that we're looking at is looking at non-viral delivery of genetic information, to these autologous therapies. So really, what other technologies can be used in lieu of LV, which can be a costly raw material to use in the process, and looking at other means to really have the genetic information delivered to those cells, but maybe in a more efficient manner or in a maybe more cost-effective manner.
Okay, well, switching to the downstream side, you know, you mentioned that you have your analytical testing services lab, which you launched last year. And what are you doing to improve the performance of your assays to characterize potency, purity, and other important product quality attributes?
Yeah, so we look at this in a few different respects. One, really want to establish the foundational assays that are just required to develop and release a product. But we understand that we're often limited in the type of data or the amount of data that you can generate on a product to then really understand the attributes of that product and how it's performing through any given process. And so we do look forward to advanced methodologies to be able to further characterize or understand our products. One of those being next generation sequencing. So in this respect, we have invested in both short read and long read technologies. So we do have the Illumina MiSeq. We also have the PacBio, and we believe that those are powerful tools to then understand both in the nucleic or in all the nucleic acid, the viral vector, and the cell therapy space. In addition to that, we've implemented sort of next generation PCR technologies like digital droplet PCR and digital PCR. So we think that those are really powerful tools that are often used to both titer your viral vector, for example, but then also be able to quantitate some residual impurities that exist in your process. And we believe that those are powerful tools that can really reduce the variability of what you're seeing, typically using qPCR-based methodologies. The last one that I'll kind of touch on here is really investing in expertise on potency assays. So depending on the type of therapy you're developing, potency assays can be quite challenging, right? Developing the assays and then really understanding and correlating the readout of those assays, to what you may see in the clinic. And so we've really invested in both the expertise to be able to develop those technologies and the technologies used in developing potency assays. And so there's just some of the areas that we've really been focusing, investing in on the analytical front, but really, taking a step back, it's understanding how can we improve upon and invest in technologies that better characterize versus, what the industry is used to right now to develop and release a product.
Okay. Well, you mentioned earlier you've designed your facilities to be highly digital. You mentioned your collaboration with Autonomous to further this objective. Another thing we continue to hear a lot about today is the potential of AI. So where do you see the implementation of AI-specific applications could also further improve the efficiency of your manufacturing processes in the future?
Sure. So, AI is very interesting because it really allows for sort of a next level, data analysis to then generate novel insights. Oftentimes you have, data that exists in disparate areas, and it's often difficult to be able to, bring those various data sets together and be able to analyze them in a meaningful manner. And so, the opportunity I see here, kind of exists in a few different buckets, for example, materials, equipment, process, and analytical performance. So, from a materials or product perspective, maybe if I'm talking more, broadly about supply chain, I think you could really implement AI to understand what your risks are or bottlenecks are in your supply chain and understand your usage or your, start lining up your supply versus demand, both in the materials you need to manufacture your product, but then also on the back end for your product as well. Another application is for equipment. So oftentimes understanding the performance of equipment is key to then maintaining the consistency of a process and understanding how that drifts over time, or what risks can be predicted through understanding current data sets, I think is important. And then that can also inform the maintenance of that equipment. Me being a CMC guy, I'm always focused on process and analytics. And so I think you can also apply this in both of those arenas. So from the process side, we start looking at chaining together the different unit operations. It's important to understand how should a process behave, not how it's necessarily designed, but how is it behaving relative to its design, and then informing either data that suggests that a risk could be present in the future, or informing how optimizations can be made to your process to then really have either improved more performance or more robust performance. And then I think another key aspect is on your analytical side. So ensuring that your analytic is performing consistently over time, and then understanding any trends in the various analytics. In many respects, these are novel analytics. These are analytics that are being used in a more robust manner for the first time. And so I think it's important to be able to understand the performance over time. And I think AI-based technologies can really help shore up or understand that performance. But really overall, it's the control strategy. It's really how do you using AI to then inform risks and improvements in your overall control strategy.
So let me ask you a different type of question, but how is being situated in Philadelphia's emerging Silicon Valley proving to be an advantage as you build out CBM?
Sure. So we think that there are multiple advantages in being in Silicon Valley and being in the King of Prussia area. So first, we think location is key. So we're in close proximity to both numerous clients in the area, right, academic institutions, treatment centers. And so we think being physically located in this area really helps facilitate a lot of the relationships, a lot of the client services that we're looking to support. In addition to that, we believe we're sort of in a hotbed for talent. So when we talk about talent, in pharma, but then extending that out to the advanced therapy space, talent can be difficult to source, and we believe that we're in the right location to be able to source talent who have previously worked on advanced therapies, ranging from nucleic acids, cell therapies, viral vectors, and so on. From a logistics perspective, we do believe we're in an ideal location, given the transportation network. So we're in an area that has access to multiple modes of transportation, and we're also on the eastern seaboards, which optimizes our access to Europe. And the final thing I'll say is, the area is #1 for NIH funding in the advanced therapy space, and so really just adds that additional layer to help support being here in close proximity to the academic and client institutions.
Okay, well, let me ask you a final question and perhaps give you an opportunity to summarize some of the things we've just discussed. But how is CBM looking to differentiate itself as the preferred CDMO partner?
Yeah, so we believe that in the CDMO space, there's really an evolution of catering the service to what the client and what the patient needs. And here at CBM, we're really trying to rethink that. So we're offering an improved service, an improved offering and experience to our clients. We believe, to start that out, establishing the right digital backbone is critical. Oftentimes, starting out implementing, a digital approach is far easier than retrofitting an existing operation. So we really took that digital first approach to ensure that we have access to our data, that our clients have access to our data, and that we can respond to that. We also have enormous capacity. So, as I said before, we're building out, greater than 700,000 square feet. So we really be able to support, many different players in the space in terms of the capacity that they're looking for. We believe that we've built a best-in-class team. So we've really sourced our leadership and our SMEs from areas where they've demonstrated success. We believe in a flexible way of working with our clients. So we don't try to fit one client into one way of working. We believe that clients are looking for different types of relationships and really want to complement that relationship. And then finally, we offer that end-to-end offering. So you can work with one CDMO to develop your product versus having to manage multiple CDMOs, which we know can be challenging.
Okay, well, great. Well, really appreciate you joining us for this session. Also, Avi will be joining us on our panel discussion coming up in about a half an hour. So we look forward to that as well. So thank you very much.
Yeah, thank you for having me.